How did you find the mock test?
You put all of our names in it.
Yeah.
That's what I'm saying. I made an appearance. That was question 1. She's question 4.
I was like, wow, I'm actually in clinic. This is probably the closest I'll be getting to med school.
I isolated a transcription factor.
Was it challenging? What?
It was. Let's just say we need to study the details more.
I haven't finished my prep yet, so I'm not gonna do all at all.
It's okay. I'm waiting for the end of the stuff today, but I'll likely the online quiz and then the quiz that you guys wrote today. I'll probably just be posting it.
Yeah, that's good.
Like, what percentage of course analysis do you get at the end of it? Is it above 50?
It's just above 50 as of this morning.
Okay, that's good.
It's not bad.
got
I was kind of hoping secretly for about 65 or about 2/3 because there's no way I can get more than 2/3. But it's not bad. So you can probably post one more time.
Yeah.
right at the end.
I'm going to keep my word. I said I would not post anymore.
There's 6 of us.
Your usual style.
Give us half the question.
Maybe. There's a lot of writing on here. Of course, vowels. How much information you have when I give you the
Depends
information.
on the percentage of course vowels.
True.
Actually, there's quite a few questions on the Facebook group already. So you'll let me know when we're switching.
So there's a slide that goes through next-gen measurement sequencing. It talks about the timeline if you do whole exome sequencing. After that, it's just a really bad segue, but we use these techniques.
So I was going to start off by saying, like, I'm going to talk about LFS. And, like, genetic testing is important and beneficial to these
Yeah,
individuals.
I saw you.
Oh, you read that? That's the exact quote?
Are you serious? Do you think you're gonna do it? Are you sure? I think I'm good. Hope so. If I pass out, you have full permission to take over. It's okay, thanks. I'm good, thanks. You need to take a break. We'll see how it goes. Okay, folks, if you don't mind, I'm going to try to get going. Um, so we have a lot to cover today. This is the last official lecture that you have for your upcoming final exam, and as you know, your final exam is actually quite soon after the end of the semester is done. And on Friday, we are going to go through a full review, and we are going to troll you a little bit, give you some ideas about what kind of questions to expect. Some of them might be identical to what you get on your final exam. And so it's actually worthwhile if you can drop by on Friday to drop by. I also wanted to just bring to your attention, we had spoken several times about the controversies in science, that sometimes science, even if it's published by reputable scientists, even if it's in the greatest journals that you can find, that sometimes there's a lot of controversies sort of surrounding it. And one of the papers that came out fairly recently at the very end of March, and then it was commented on today, is related to one of those molecules, GDF11. And we spoke about GDF11, the anti-aging factor, and it's a factor that you should maybe go back and review a little bit for your final exam. It's related to exercise, it's related to anti-aging, and there's still controversy about it. In fact, the two groups, in an unprecedented sort of commentary, both groups came out in the same journal article saying why their results were actually correct. So you have two different sets of results, actually completely opposite to each other, and you have one group saying why their results are right, then you have another group in the same journal saying why their results are right. So science is one of those things that all of you like to think that it's sort of set in stone, that we have all the answers to all these different types of things, but the reality is we really don't have all the answers to everything. Now, we want to, in this course, also talk a little bit about some of the technology that's involved in the different types of results that we get in these lectures. And so one of the things that is important to you, as I mentioned at the end of the last lecture, is this idea of personalized genomics. Ultimately, down the road, when you have an infant going in and they're born, they're no longer going to have the little heel prick test. They're going to actually get a full genetic workup or a full genome scan done. And it's probably going to happen in your lifetime. It is now efficient as well as being affordable to be able to do personalized genomics. And it's no longer in the realm of science fiction. And coupled with CRISPR-Cas9 that we talked about earlier, your lives will be heavily impacted by this idea of genomics and genome editing. And along the way today, we have actually, your TA is a specialist in a type of cancer called Leaf-Remeni syndrome, or LFS as abbreviated on that previous slide, who's going to talk to you about the impact that personalized medicine, personalized genomics, genotyping, genome screening actually has here in Toronto in terms of different forms of cancer related to Leaf-Remeni syndrome. So I'm going to spend the first few minutes, and again, I have to apologize that some of the slides from the last lecture I had not loaded the correct slide deck. And even though I went through the lecture, they didn't really jive well with what you needed to know. So I wanted to go back and talk to you a little bit more about some of the different polymorphisms. They're not always single nucleotide polymorphisms, but changes in the DNA sequence that we can understand related to depression. And I had left you with this 5-HTT-PLR sequence, both the long version and short version, and I'll go back over that today, and then talk to you about how we actually go about looking at SNPs. All of you have heard that our DNA, every one of us has a unique sort of DNA fingerprint. And how do we actually get to the point where we can actually say that all of us have unique DNA fingerprints, and what does that actually mean? And I want you to have some ideas about what that means by the end of today's lecture. So I had left you sort of with this bizarre concept that here in the 21st century, that if someone like myself had depression, there's really no way for you to tell if you're sitting beside someone with depression or if I have depression, etc., to know that someone has depression. The best technique that we have right now, there's no brain scan, there is no blood test, there is no way to draw blood and see that your enzyme levels have changed, therefore you have depression, unlike in colorectal cancer. So right now, our best diagnostic criteria is for you to sit in front of someone and have them ask you a bunch of questions, and then for them to make a judgment call as to whether or not you have depression. We know and we think that there are changes in your neurotransmitter levels. It might be a change or a decrease in dopamine, or it might be a decrease in the serotonin levels in your brain, or it might even be a change in your noradrenaline levels in your brain. But overall, these monoamines, together these class of neurotransmitters known as monoamines, we think that they change in an individual that has depression. It doesn't mean it's the cause that you have depression. It just means that overall there is a change that we can see. And all of you know, or many of you will know, that one of the therapies that we have is to actually try to reverse that phenomenon. If this low level of neurotransmitter is what we think is involved in depression, well, if we reverse that, remember that idea that we try to fix something that is broken in terms of the types of medicine that we practice in the West. If we do that and we can reverse sort of this trend, maybe we can actually reverse depression. And so this idea that maybe neurotransmitters are the key functioning element in depression, it becomes an important diagnostic criteria. So we're not really satisfied as biologists with that because it's really unsatisfactory. So how do you know if you have depression? Well, you don't know. You have to go and visit a psychiatrist who has to interview you, and you have to talk about it. There's no brain scan, blood test, or anything else that we can use to actually identify whether or not you are at risk for developing depression, whether there's a genetic element to depression, etc. And so this Caspi paper that I mentioned to you in 2003 was really important because it identified a polymorphism, a change in your DNA that allowed individuals to say with some certainty in this 2003 paper that was published in Science, to say that if you have one version, the long version, you are protected, and you have the short version, you are at increased risk of developing depression if, and only if, there is a stressor in your life. Not a guarantee, but at least it's a way to quantitate and to show that there might be a genetic element to major depression. So just a couple of points. The type of polymorphism that we're talking about here is not a single nucleotide polymorphism. It is actually a rather significant amount of DNA that is being changed or lost or deleted. And we'll talk a little bit more about that. And it occurs here in the promoter region, as I mentioned to you before. It is not a structural change in the transporter itself. Your transporters in someone who has the short version are identical to the ones that are found in individuals that have the long version. And overall, in terms of chromosome 17, the only reason that we can actually detect this is because people were looking for these changes related to serotonin, and this is the gene that encodes for a serotonin transporter. So if you have the short version, where you have this smaller promoter region, that means that overall in the 5' region of this promoter region, you are going to produce less of these serotonin transporters, which means that you probably have less serotonin in your brain to begin with. So this was a very unusual study. It's not the type of study that we normally have in science. It's one of those prospective studies. We have a hypothesis and then we move forward and test this hypothesis after about 10 or 15 years. Can we actually see a genetic influence and the resulting depression as a result of this prospective type of study? But it was a remarkable study in many ways because it actually suggested that maybe there is a genetic way to actually go about looking to see why maybe one individual has depression and another individual doesn't. And this is sort of the promise that personalized genomics or personalized medicine has. We know that we all have a large number of genetic variations in our DNA. Some of them, and in fact, it turns out that the vast majority of them, the increase in frequency that you see here, the bigger the pie is, the more likely you're going to see these types of changes. And we get lots and lots of little changes, single nucleotide, one base pair changes. We have lots of these. We have millions of these. You're sitting there right now in your DNA, compared to your neighbor, you have millions of these single nucleotide polymorphic changes of one base pair. And we have lots and lots of these. And these single nucleotide polymorphisms, as you'll see on the next graph, in relation to this slide and the next one will be really important for you on your final exam. So we also have other things. We have these small changes
bigger than one base pair, so a little bit bigger. It could be like 1 kilobase of genetic information that's missing, or it could be less, somewhere in this continuum. And one of the things that we could have are similar to what we saw with those SNPs, with those polymorphisms associated with major depression. We might have an insertion where we add DNA to our sequence. That can certainly happen, and that's called an insertion. Or we could have a deletion, which is what we just saw with the short versus the long form of the 5-HTT PLR. So we can have these changes as well, and these microsatellites or indels are really important as well, but they're also fairly common. We have a lot of these in our DNA as well. However, once we get into bigger structural changes, where you're removing or changing large chunks of DNA, we're talking about structural variability in our DNA. And I'll work my way from way down here. It is very unlikely that you are sitting here in this lecture room right now that you have a large structural variation in your DNA. It means that there's been a translocation event. You have a large-scale genetic mutation, and it's very, very unusual. It's very unlikely, and it's, in terms of frequency, it's very, very small. And this is where we see large portions of our DNA, megabases of DNA, being rearranged. So they could be a translocation, etc. And again, you'll have to take this slide with the next slide for your upcoming midterm. And again, everything in between, you can see that generally speaking, we have lots of small little changes, but big changes to our DNA are not really frequent events. They're very, very unusual, very rare events that we see. So how do we actually go about looking for these different types of variation? And here is where I want you to be able to match up what we see with changes in our DNA with the types of things that we use to measure these changes. So we had also talked about karyotyping and fluorescence in situ hybridization very briefly. We've spoken more about karyotyping, particularly in relation to the Zika virus, and we knew that, for example, for some forms of microcephaly, that karyotyping actually might be a way to see this type of large-scale chromosomal rearrangement in an easy fashion. When we're looking at these intermediate changes down here, where we're looking at changes that are somewhere between 10 kilobases to 1 kilobase, we can use microarrays where we have spotted onto our chips. We have a number of different DNA sequences that can be hybridized and allow us to know through microarray really quickly if we have these larger scale changes. In addition to that, what we're going to be talking about here today, when we're talking about genotyping and when we're talking about sequencing and we're talking about whole genome sequencing, we're talking really about the actual word sequencing. We're looking at your individual string of letters on single nucleotides, and we need to have those single nucleotides, otherwise we wouldn't be able to detect a single base pair change. The sequencing is much more sensitive than a microarray is. This is looking at a whole stretch of DNA. It's just looking to see if, for example, is there a building on campus between St. George and College Street. Here, at this individual level, we're looking for a specific individual in a building inside along that stretch of road that we're looking at. So it's much more specific, and as you can imagine, it's much more time consuming. In fact, it was one of those things, once we got really good at this, back in 2007, this was considered a scientific breakthrough of the year, because we realized fairly quickly that if we did this the right way, that in theory, we would actually be able to use this as a fingerprint for your future health. So you may not be sick right now. You may not know that you have a genetic predisposition to a particular drug, because you'll maybe never encounter that drug until you need to have that drug. For example, some of you might have heard of a drug thinner called warfarin. And there are genetic polymorphisms that make you more susceptible to bleeding longer if you're given warfarin than if you don't have a single nucleotide polymorphism related to that. So how would you know? Until you actually try and have warfarin, you'll never know that you have it, or you're susceptible to bleeding out if you're given warfarin until that happens. Unless you know your genotype ahead of time And your doctor, just by looking at your genotype and all of your single nucleotide polymorphisms, can tell you that, oh, you know what, you should not take this drug. You're going to have a reaction to this drug, or if you take this drug, it's going to cause an adverse drug reaction. And this was sort of the promise that was put out there by GWAS studies back in 2007. And so the idea is, if we take a look at everyone's DNA, and we know that there are many, many changes and differences between all of your DNA in here, and we have a lot of these single nucleotide polymorphisms. Remember the far end, the right-hand side of that sort of uphill graph? We know that we have a lot of these variabilities. Sometimes those variabilities mean absolutely nothing, has no consequence whatsoever. It's just a different way to code for the same amino acid. But sometimes those changes might be really important, both in terms of health as well as disease. So you might have one SNP at one position on your gene. Your neighbor sitting right beside you might have another SNP or another single nucleotide polymorphism, either a G or an A, that may or may not have a functional consequence. And so one of the things that the genome-wide association studies does is it wants to compare all of the genomes from a number of different individuals that you see here. If this is a normal population, we would be able to see that some of these individuals in this population have this particular SNP here at the top, and the other individuals in a normal population without a disease condition, for example, would have all of the other SNPs that you see down here. And what you would do is take all of these individuals in the normal population, all of those individuals in the disease population, look at all of their genotypes, and see, by using statistics, is there a genetic trait? Does that single nucleotide polymorphism actually make you sick if you have this single nucleotide polymorphism, or make you more susceptible to being sick? And so this is sort of the whole idea or the rationale behind personalized medicine. And again, what we're looking at is a control or normal population, and looking at all of their different single nucleotide polymorphisms, their entire genetic makeup. Looking at a disease population, for example, we might compare normal individuals versus individuals that have autism. We want to look at any of the SNPs that might be associated with autism and see if there's a difference between the group that has autism versus the normal control population. We would then put the DNA, hybridize it to a SNP chip, which allows you to very quickly look at the sequence. And what is generated for each of the different chromosomes that you see down here, including the X chromosome, is that you'll see all of the different single nucleotide polymorphisms across both populations. And one of the things that will jump out at you, if you were to take a look, is that when you're looking at two different populations, a control versus a disease population, some of those SNPs seem to be more closely aligned with the disease population than it is with the normal population. And so if it reaches a certain level of statistical significance after a statistical correction known as a Bonferroni correction, on this type of plot that you see here, which is called a Manhattan plot, and it looks like a bunch of the New York Manhattan skyline, and you see all sorts of different heights of buildings. If you take a look at this, you'll see that some of these are really quite statistically significant. And those are the particular SNPs that you would go back and you would test based on this Bonferroni corrected Manhattan plot to see if there's an association between that SNP and that particular disease condition. Again, I'm using autism sort of as a model for this. Do you guys have any questions about this before we go on? Okay, so if you're looking at both the maternal-paternal set of chromosomes, we have about 10 million common SNPs in our genome. So I have 10 million, you have 10 million. Each of you have millions of different SNPs. And what we would have to do, there's no use in doing it one-to-one. So for example, I know I have a lot of different SNPs in my genome. You know that you have a lot of different SNPs in your genome. But that just isn't enough. Like, in order for us to be able to tell, like, the reason why Dr. Ju, for example, tends to be follically challenged is because he's missing these SNPs here. it wouldn't be enough to be able to do that from one sample to another. So in order for us to determine it's not just unique to me, but this is the gene that causes follicular challenge, one of the things that you would have to do is actually look at the genome for thousands of different controls, as well as 1,000 different case studies, and look at everyone's genome at the same time. So we'd have to genotype all the DNA, look for all the different SNPs, and this means that in theory you would have up to like 20 billion genotypes that you're looking at. So obviously there's a lot of number crunching, there's a lot of statistical analysis like the Bonferroni that goes into place. So this sounds really ridiculous in terms of these numbers, right? So in 2002, so more than a decade ago, once we were fairly good at this, if you were to do massive amounts of genotyping, actually for a particular genotype, and we're talking about a genotype, not genome sequencing, but for looking at one genotype, do you have like type 2 diabetes, the gene for type 2 diabetes, and we looked at one genotype for one disease, it would be about 50 cents. So if you didn't know this and you're just taking sort of a randomized approach, I want to just know what the differences are. If we just went through every last known disease with every last known gene, it would roughly be about $10 billion. No research science would ever be done in terms of looking at this. If you look at 20 billion genotypes and you're looking at 50 cents a genotype, that's about $10 billion for each disease case. Nowadays, though, it's become much more efficient. It costs less than a penny to be able to look at a genotype. It may not sound like a lot of money, but it actually is a tremendous amount of savings, and we're actually even better than this right now. So again, I won't go into this in a lot of detail. It's really just to show you that we traditionally start off with a hypothesis model where we're looking at a genotype in a small number of individuals, then we scale it up to a larger number of individuals in this second stage, and then in the third stage we'll actually go back and check to verify whether or not this genotype that we looked at is true in a different population, for example, or in a larger population, just to verify that this SNP that we see is indeed accurate. And this idea of replicating this because of the large numbers of variation that exist within our genomes is actually a really important phenomenon. So again, I won't go into this in a lot of detail. What I'm going to be talking to you about today is really about understanding, boy, you know, your cough sounds really terrible. I hope it's not as bad as it doesn't hurt, does it? No? Okay. Thanks for trooping through the lecture. So when we're talking about this model of DNA to RNA, right now actually the most exciting stuff isn't in the DNA. The most exciting stuff is actually in the RNA. So I'm hoping that down the road in your third and fourth year courses, after this course is over, when you talk about the most exciting part of molecular biology, it actually resides here at the RNA level, not necessarily at the genomic level. But I want to talk to you a little bit today about why we actually have to know these different DNA sequences and how that actually is of use to us. And I'm going to use two different disease phenotypes that are associated with cystic fibrosis. And cystic fibrosis is this disease here where you accumulate fluid in the lungs. Usually it tends to be very, very severe depending on the type of change that you have in your DNA sequence. And one of the things that we know and we can map out is that if you have problems like this young individual over here on the left has, is that we have mapped things out fairly early in the 1980s to the cystic fibrosis gene on chromosome 7. But in addition to that, in the cystic fibrosis gene, one of the things that we realized is that even though the disease itself revolves around having some sort of alteration in the cystic fibrosis gene that we isolated, there are many, many different types of mutations or single nucleotide polymorphisms that can actually occur. So normally if the gene is doing its job, one of the things that happens is the DNA gets transcribed, and then the RNA gets translated, and then it forms this chloride conductance channel, which allows chloride to go in and water to leave. or vice versa. So it's a channel that works in both ways. And so one of the things that happens is that your lungs don't get filled with water because of this particular channel. However, there is a mutation that occurs called the delta 508, and what happens is that this channel actually gets, the DNA is there, it gets transcribed, but one of the things that happens is that it doesn't work its way up to the cell surface. Okay, and it gets trapped inside the cell. So the channel actually can't do its normal job. It can't conduct chloride. It can't effectively remove water, and so you get a buildup of fluid in the lungs. There is another mutation, though, the G551D, that actually allows the chloride conductor to work its way up to the surface. They still have problems with fluid buildup in their lungs, but it's a totally different scenario. It's the same gene. It is a different scenario. It involves a different position on the gene, and this is the reason why we actually have to genotype. We need to understand the genetic sequence because this clinical situation where we have G551D is very different than the delta 508. We can actually help this individual with different drugs on the right than the drugs that we would use with the individual on the left. So imagine, for example, you had the delta 508 mutation. And one of the things that happens is you can't really breathe while you start accumulating fluid in the in your lungs. And this drug treatment that you're given, you're either given a choice of lumacaftor or Calideco, and depending on the type of mutation or single nucleotide polymorphism you have, the drug is either going to work really, really great for you or it's not. So if you have the delta 508 and you're given Calideco, it's not going to work for you. If you have the G551D and you're given lumacaftor, that's not going to help you breathe any better. But if you are with the 508, you've been genotyped, we know what type of DNA mutation we have, and you're given lumacaftor, this actually will increase the trafficking of this misfolded chloride transporter up to the cell surface. This drug here actually works to keep the channel open longer. And so one of the things that happens is that this channel is there, it's in the right location, it just doesn't stay open as long as it should. And this drug actually works to keep that channel open for longer periods of time. It can't do that unless the channel is actually there, though, so it wouldn't work at all in this particular scenario. So knowing the genotype is actually really important for us to be able to prescribe the right sort of treatment for this individual who has exactly the same symptoms of having fluid buildup in their lungs. Now again, I won't go into a lot of detail. All of the modern techniques that we talk about when we look at sequencing, every last one of them actually still revolves around this idea of using polymerase chain reaction. So whether you're talking about a new technique called next generation sequencing, or you're talking about older techniques like the Sanger sequencing, all of them make use of the fact that DNA can be amplified, and it requires that you have the appropriate DNTPs or nucleotides, as well as primers, and the ability to actually amplify the sequence over and over again. Have most of you heard, you've all heard of these different techniques, right? Northern blotting, southern blotting, westerns. So can you quickly tell me, what do you actually measure with northern blotting? What molecule? Okay, RNA or mRNA in some cases. Southern blots are used to measure DNA, and then Western blots are used to measure protein levels, right? And RT-PCR is really good for looking at or expanding different DNA sequences. And all of these things are really good for different types of things. You can determine like overall expression levels, and you can compare for the expression of RNA for northerns or for proteins using Western blots. However, these techniques are not really great for small variations. Generally speaking, even with RT-PCR, finding out if you have like a one nucleotide variant, it's not really good at being able to detect those particular changes. And so this gets into this idea. Way back in 1977, when PCR became more and more affordable, one of the things that people wanted to do was to use this method called Sanger sequencing, which allowed this piece of DNA on 4 different wells, the same piece of DNA to be amplified using particular nucleotides that we would be able to track. And so allowing for different size fragments of DNA to be run on a gel allowed us to go back in after doing this primer extension on this Sanger sequencing model. If this is my template, this is the DNA sequence that I want, and I know that in this lane I've amplified for A, and in this lane I've amplified for T, C, and G. Because they are running at a certain size and the smaller fragments are down here at the bottom, simply by being able to read from the top to the bottom, I would actually be able to go back in and recapitulate the entire sequence of this DNA. And these were sort of the techniques that were used way back in the 1970s and into the 1980s. I had to do that when I was in grad school. It was a lot of fun in grad school way back in the 1990s to have to do manual sequencing, because you could loaf off a lot. You could say to your supervisor, you know, I'm going to set up a sequencing reaction, I'm going to let it run, disappear from the lab all day, and then come back in the evening, and then do the rest of your experiment, and then tell your supervisor the next day, I'm going to spend the rest of the day trying to go back and read the sequence, so I'm going to go to the library for about 4 or 5 hours. Nowadays, you can't do that, unfortunately, so you actually have to be in the lab to be able to do your types of experiments. So that's the bad part of, I guess, being in a modern sort of lab. Again, nowadays we do exactly the same thing. All of them are based on the same idea, but we try to automate things as much as possible. So we use this dideoxy sequencing. We have little fluorescent tags on the different nucleotides, and as they get incorporated, we can actually see different fluorescents being added to the particular sequence, which allows sort of a laser beam as it moves across to be able to read the particular sequence as it goes along. And then someone had this really bright idea. You know how your cell phones, actually you guys are doing it the exact opposite way. You remember a few years back, cell phones got smaller and smaller before now they start getting bigger and bigger again. So way back in the day, someone had this idea. Well, you know these gels, because I don't know if you know what size these gels are. I used to run 3-meter sequencing gels. Do you have any idea how tall 3 meters is? So it's a little taller than me, so you'd have to go up on a ladder, you would cast a gel, and then it's like 3 meters worth of reads as well as everything else. Someone had this great idea. You know, you can't run very many of these 3-meter gels at the same time. Why not make these gels like the size of a capillary tube? And so people started to figure out ways to make these more and more compact, which allowed for faster and faster reads. And all of the next generation sequencing data now goes back to exactly the same thing. You do these same experiments, the same sort of sequence reaction over and over again in parallel. So instead of just running one gel with one reaction, you run like multiple sort of spots or multiple locations, and you do the same sort of thing. And now, instead of being able to do this multi-year, decade-long study that involved multiple labs that cost over $3 billion, you can literally get this done now overnight if you're doing whole exome sequencing at the cost of less than $600 for whole exome sequencing if you send it out. So again, next generation sequencing, the only things I'm going to mention about it is that it's high throughput. And this is a slide I'd like you to make sure that you are familiar with. So it's high throughput, millions of parallel sequences going on at the same time. We're now at the level where we can do your entire genome, every single nucleotide base for less than $1,000. We're not really sure yet if the quality is as good as when I used to do Sanger sequencing way back in the day, but we kind of hope that it is just as good. And again, nowadays, what we do is we take your DNA, we do capture if we're doing whole exome sequencing. The sequencing itself only takes 1 to 2 days. We have to align the sequence to a reference sequence, which takes some time, and then we have to look for individual variations. You have like 5 to 10 million different changes. I have 5 to 10 million different changes. You have thousands of individuals, and these variants, and calling these variants actually takes a fair bit of time, even though it's an automated procedure. Then you actually have to go and see if they line up to any genes, which is what this annotation is. And then you have to try to figure out, do those actually mean anything? So what if these variants are occurring at introns? Do they actually mean anything? Probably not, but actually nowadays we're not 100% sure. If we have these changes at intronic levels, we also think that this might be important as well. And so this gets into sort of the next stage. You get all this information, and I mentioned cystic fibrosis. What does this actually mean for us here in Toronto? And I think that's yours now.
Can I make this smaller? Oh.
Yeah, I have a big head.
And then how do I do this?
This is this. And you need the microphone. You can just put that in or carry it around. She looks a little nervous. Why don't we give her a hand?
Aw.
Anytime you're ready.
Can you hear me? Okay. Okay. Okay, so today I'll be talking about Lee-Fermini syndrome, and it's also known as LFS. Wait, can I start again?
Who's grading Zeta?
Ha, that's fine. Okay, so today I'll be talking about Lee-Fermini syndrome, and it's also known as LFS. So LFS is one of the most well-recognized cancer predisposition syndromes, so individuals have a high risk of developing cancer. It was first recognized in 1969 by doctors Fred Lee and Joseph Fermini. They conducted epidemiological research, and they reviewed over 600 patient records, and they noticed a pattern within specific families. They noticed that in these families, individuals were developing tumors at a really young age. So adults were developing tumors 20 years younger than when you would typically see these tumors arise sporadically in the general population. Also, in these families, children were developing tumors within their first decades of life. They also noticed that in these families, they weren't even developing one type of tumor. They were developing a broad spectrum of tumors, such as breast cancer, sarcomas. So sarcomas are cancers that arise from mesenchymal cells. They were also developing either tumors of the brain or the adrenal glands or leukemias. What they also noticed was that in these families, there's an autosomal dominant pattern of inheritance. So this means that the affected individual that has cancer, they have a 50% chance of passing on this predisposition to their children. So about 20 years later, in 1988, Lee and Fermini come up with the classical LFS definition, which is the individual that you're interested in, if they have a sarcoma at a young age and they have two close relatives that also develop a cancer at a young age, they likely have LFS. Also, what's interesting is that if you develop a tumor when you're young and you survive it and you have LFS, you have a 50% chance of developing another tumor in your lifetime. So as you can see here from this pedigree, the shaded shapes are the ones that are the individuals that are affected with cancer. The child at the bottom, they developed a rhabdomyosarcoma when they were 3 years old. So this is a soft tissue sarcoma. They survived it, but then they developed another tumor at the age of 9. So what's going on in these families? Is there a gene involved that's causing all these cancers to develop? Well, back in 1969, DNA sequencing wasn't available, so you could not test potential candidate genes. But let's fast forward to the late 1980s. During the late 1980s, the role of p53 is emerging. So p53 is a protein that functions as a transcription factor. It flies around the cells and looks for DNA that is damaged. Why is it looking for damaged DNA? Well, damaged DNA can cause abnormal cell growth and cause the cells to become tumorous. So P53 is functioning to prevent tumors from growing. So we call this protein a tumor suppressor. It also prevents tumors from forming by causing cells to die, programmed cell death, and this is known as apoptosis. So what does P53 have to do with LFS? Well, again, in the late 1980s, a young Canadian, his name is Dr. David Malkin, and he's currently my PhD supervisor. He did his MD at the University of Toronto. He completed his residency at the Hospital for Sick Children. Then he goes to Harvard, and he joins Dr. Fred Lee in his in his clinic to learn more about childhood cancers. But David Malkin falls in love with science, and he decides to pursue a postdoc. And then he has this hypothesis: is P53 involved in Lee-Fraumeni syndrome? Well, his hypothesis is correct. Indeed, in Lee-Fraumeni syndrome families, the P53 gene is mutated in the germline. And germline are sex cells, the cells that are passed on from generation to generation. So based on this information, you have a family history of LFS. This P53 gene is associated with LFS. What do you think we should do? So say you have LFS, you find out your P53 gene is mutated. What would you want to do? Yep. Yes, exactly. So say I have LFS, I find out my gene is mutated, I would want to start screening to see if any tumors are forming in my body. So that's what we do. So you have a family history of LFS, you have inherited the P53 mutation, you start screening. So based on your specific tumor, there's different screening protocols, and these screening protocols were developed at the Hospital for Sick Children, and is now known as the Toronto Protocol. So this includes annual MRIs. For example, this child here, she comes from a Lee-Fraumeni family. She had P53 testing, and they found it was positive for the mutation. They started conducting MRIs, and they found early onset tumor formation. So tumors detected, you start treatment. So treatment for cancers includes surgery, radiation, and chemotherapies. Surgery may not always be feasible depending on the site that the tumor is found. So you can treat the cancers with chemotherapies. So what do you think the survival rate is of individuals who have LFS and they start screening? Do you think they have a greater than 80% survival rate after screening? Mixed nods.
What's the survival rate without screening?
So what do you think the survival rate would be if you did not start screening? Okay, well, the study was conducted in my lab. We had individuals with LFS and the TP53 mutation, and we asked if they wanted to undergo surveillance, so start screening for tumors, or if they did not. 18 individuals decided to be put on surveillance, and 16 did not want to be put on surveillance. Over time, in the group with surveillance, 7 individuals developed a tumor, but they were asymptomatic, so there were no signs that a tumor is forming in their bodies. They didn't have any pain or anything, but due to screening, we found the tumors really early. And they should say 3 years later, but 3-year overall survival was 100%. All 7 individuals that developed asymptomatic tumors survived. But in the other group, 10 individuals developed a tumor, but since they weren't put on surveillance, by the time the tumor was detected, it was at a high stage and a high grade. Unfortunately, only 2 out of the 10 individuals survived, so the overall survival rate in the non-surveillance group was only 20%. So this shows the importance of starting surveillance. So to summarize, LFS is rare, but it serves as a paradigm for studying hereditary susceptibility to cancer. And it's a great example of how we can apply our ability to integrate really interesting research questions. It's also a great example of how clinical observations can lead to really interesting research questions. So the clinical observations that these families are getting early onset tumors led to the research, and the pattern of inheritance, that is autosomal dominant, led to the research questions, are genes involved? And then when we found that the P53 gene is associated with the syndrome, that led to further redefining the syndrome. Also, this P53 information led to further management of the disease. You can get genetic testing, be put on surveillance, and it can increase your overall survival. So that's all. Any questions? Yep.
Can you just clarify what LFS is again? Like,
Okay,
what
so
exactly defines you have your-
okay, so the classic definition is that you have the individual you're interested in. So say the patient comes into your clinic, if they have a sarcoma, so a tumor that arises from mesenchymal cells, and then you look at their family history. So based on the classic definition, if they have 2 family members that also have a tumor, any type of tumor, younger than the age of 45, they're likely to have LFS, and then you would do the P53 testing.
Okay, so folks, what we're going to do on Friday is what I'm going to ask you to try to do, either on the discussion board or through your Facebook groups, is to come up with any slides that you want to be explained in more detail. I am going to give you a full review based on what we did for your midterm, so which lectures you should concentrate more on, what sorts of concepts you should concentrate more on. Whenever you see any of the different diagrams, by the way, as you heard in tutorial today, you should really make sure that you understand them. So if there are certain ones that you want to go over again on Friday, because the time to Friday to your Tuesday actual final exam, it's really, really short. So I'm a little concerned, to be honest. So I want to make, or is it Wednesday that you write your final exam? Wednesday, sorry. You get an extra day. So I want to make sure that you're well prepared for it. And the only way to do that is to use some of Friday's question time to actually be able to answer any questions that you might have. We will also go through some practice questions on Friday as well that will give you an idea of what you are either lacking or what you need to study some more. I hope to see you Friday and have a great week. And make sure you go to tutorial if you haven't done so already.